Professor John Scott. I'm the Edwin G. Krebs Spates Chair of Cell Signaling and Cancer Biology and Chairman of the Department of Pharmacology at the University of Washington. I did my undergraduate degree in Edinburgh University, and then I did my PhD at the University of Aberdeen. And immediately upon completing my PhD, I moved to work at the University of Washington as a postdoctoral fellow with Ed Krebs and Ed Fisher. So that's my full pedigree. And then after 4 years of postdoctoral research, I moved to Oregon, and that's where I really established my own area of science, this whole field of ACAPs, and then moved back here in 2008 as the Krebs Chair. As an undergraduate working in biochemistry, my goal was always to work on human disease. I was only really interested in working on human disease. Very interested in the chemistry side, but really wanted to work on human disease. And so a sort of natural progression from that was to work on major diseases. And so we've worked in heart disease, and the logical progression was to work on cancer, particularly with the discovery of the DNAJ PKAC mutant kinase. It was a natural progression to begin to work on this, because it really allowed me to take advantage of my past experience and apply the expertise within the lab to a really important critical problem. I think that, you know, one of the things is that with research, you're constantly doing new things, you're constantly interacting with new people, and you're learning a lot. And you're learning a lot not just about the techniques, but you're also learning a lot about the people that you work with. One of the things I really like about the FLC Foundation is it's also an opportunity to interact with the patients and their families, and that's very, very motivating for researchers. Our research is focused on a very recent discovery that we think is fundamentally important to understanding the etiology of fibrolamellar. And what we've discovered is that the enzyme that goes awry in fibrolamellar is preferentially stabilized inside the tumors. And that's really important because it means that that enzyme is more active because there's more of it. And also because of that discovery, it makes that enzyme more sensitive to potential therapeutic intervention. So we think that we've identified drug combinations that can target the enzyme because it's a unique enzyme, and it's only in those cells. So from a pharmacological point of view, that's very exciting. Definitely think that some of the combination drug therapies are an important way to move forward on understanding this. I think that it's also equally important that some of our colleagues in the field are trying to develop better models to study fibrolamellar, because one of the limitations has really been the availability of, you know, accurate models to be able to perform the sort of experiments that we do that are a prelude to clinical trials. So very hopeful that what we're doing, we've already begun to talk to clinicians, will allow us to sort of bootstrap our way into clinical trials. We're not there yet, but we're definitely moving in the right direction. I think the whole field is moving in that direction. Collaboration is absolutely vital, and I think that that's one of the really unique facets of this particular field, and I think the foundation is very much should be applauded for being able to sustain and foster this very collegial atmosphere, because when people work together, you not only increase the brain power, but you increase the opportunities for innovative solutions. And I think that by breaking down the barriers of kind of competition and opening up the sort of playing field to innovative ideas and working as a team, is a very, very important step forward. It's not, frankly, something that happens in every single field of science. And I think that that's something that really the foundation has taken a lead on and really should take a lot of kudos for. I think another aspect of that that's important is the patient advocate element. I think it's very, very important. As you know, we have met with patients and met with their families, and by building relationships with them and being honest about where we're at, I think it gives everybody that added impetus to try and work towards a cure. I think one of the biggest challenges is that we know the genetic lesion, we know exactly what goes wrong, but we don't know what the cause is. And I think that one of the things that makes that more challenging is also that it's a relatively rare disease. So we think we know what's going on, but if there are more cases that come up, because disease is, you know, a very fickle thing. It's not just usually one thing, and that's what makes this very unusual. So I think that there are still some surprises that only more research are going to unravel. I think that certainly meeting with researchers and really inspiring the researchers is important. Obviously, the financial contributions are huge and very, very vital to be able to move forward, because research is a very expensive thing. But just really community with the researchers and you know, because I think to understand that both groups, the patients, their families, and the researchers, are human beings with common goals and thoughts and aspirations, really is very important to be able to knit that community together. Because when you have a community, people are going to work for each other. So the idea that a researcher knows who they're working for and a patient knows who's working for them, I think is very important. You know, I've done a fair amount of things, but that doesn't really, that's a reflection of the work that we've done, but and it's very, very nice to get that recognition, but it doesn't change things from a day-to-day basis.